So now let's talk about the use of antibiotic-resistant markers to select modified bacteria. So a big problem with genetic engineering is that it has a very low success rate. For example, in other words, a vector isn't taken up by target cells. So cells don't often take up the vector. This means that scientists have to have a way of checking whether or not the gene has been successfully transferred into the target cell. So scientists may not be able to tell if the cell has a vector in it or doesn't have a vector in it. So scientists need a way of checking which one has a vector. Otherwise, the target cell won't have the desired characteristics and won't be of any use. So one method scientists use to identify transgenic organisms is use a gene for antibiotic resistance as a gene marker. So let's look at how this is done. So the first stage is that the antibiotic resistance gene is inserted into the plasmid along with the gene being transferred. So this is the desired gene. And this is a gene that's resistant to a particular antibiotic. And these are both added simultaneously to the vector. So in the next stage, as usual, this plasmid is transferred into the target cell, for example, a bacterial cell. And the next stage is that these cells, these target cells, are grown on agar plates containing a specific antibiotic, and this usually kills the target cells. However, cells with the antibiotic resistance marker genes are resistant to these antibiotic and then won't be killed. So cells that aren't resistant to the antibiotic are killed.
Hey guys! To continue watching this video and unlock hundreds of other super concise and exam board specific GCSE biology and combined science videos, just click the Snap Revise smiley face. Join me today and together let's make biology at GCSE a walk in the park.